yeah.
I think we're ready to get started, guys. Thanks so much for everybody being here.
Okay.
I want to thank our audience for joining us today. I think we have some more people that are coming in, but I want to thank everybody for joining us for our TEN Life Science Syndicate presentations today. And today we have a special treat. We have Elizabeth Cross-Nichol of Life Science Washington Institute here to talk a little bit about how to invest in life science deals to kick us off. And then after that, we're going to have Adam Greenspan of Trace kick us off with the presentations, followed by Creative Biotherapeutics, Rejuvenation Technologies, and Scaffold Therapeutics. So I want to thank everybody for joining us today. And Elizabeth, can you tell us a little bit about yourself, and then let's jump into your presentation.
So thanks very much. I'm a physical therapist, and my background has been in ICU, working respiratory as well as neurology. And I decided that I was doing hospital administration and it would be helpful to actually do, and I did an MBA and a master's in health admin at Boston while my husband was at Harvard. I think we might have been there around the same time, Chris. And so we have here in Seattle, I'm actually now been here 20 years. My focus has been on consulting to small startups, working with a variety of small startups. We have exited and sold to distributors, and I'm in the middle of running right now, which is actually looking at reperfusion injury and doing an aerosolizer of isoflurane to prevent reperfusion injury after cardiac arrest. And I'm doing that with my mechanical engineer spouse, as well as my and the chief of anesthesia here at Harborview and University of Washington here. So I'm excited to be able to talk a little bit about how I have then got into angel investing. I believe also in being a Canadian on ecosystem development. And so I've been thrilled to be able to work with startups to be able to move them along in terms of bringing products and services to market, which I think will help everybody further down the line. And now I'm working for the Washington Research Institute, and it is actually I'm there doing grant management on SBIR, STTR matching funds, as well as on some startup funds for the companies in the state of Washington, which does not require a grant. So what I thought I'd like to talk about is what I see often when I'm consulting with people who want to be new angel investors. Mossy Ventures and Apis Health Angels are an education program. So we spend a lot of time educating people about how to do due diligence, how to look at a deal, how to decide whether or not a company is a strong team and ready to embark upon a successful move to commercialization. So I'm going to talk about the pitfalls of life science investing and how to de-risk them. I'm going to use a framework. Often people are members of the Angel Capital Association, and that is our collective international, it was actually national, but they do have relationships with international other associations that actually talk about education that's helpful for everybody. So there's a framework that was established for valuation of pre-revenue companies, and Bill Payne is often a thought leader and runs many of the academic programs. And so I'm going to use that framework as a way of talking about risks. So if we look at one way of finding out pre-revenue valuation, there's a number of factors that you can look at: the strength of the team, the size of the opportunity, product, technology, and your IP, and then whether or not you're in a competitive space, marketing, sales channels, and partnerships, what happens in the competitive environment, and the need for additional capital. It'd be interesting because different people weight the relative weights on the side differently. This is just an example. So I think we'll just talk through the strength of the team. So many people get into investing and don't really understand how complicated this layer structure is of people who are involved. So we've got Researchers and academics who are actually trying to come up with projects but may not have worked in a commercial space. We've got assessors and regulators who are actually setting the rules for what products can come to market. Payers and policymakers is another bucket of people that we think about, the FDA and the CMS, who actually make decisions on financing and whether or not a product will go to market with a reimbursement. We've got people in the life sciences industry and healthcare providers who are interfacing with patients, and then also patient engagement organizations. And as we see the evolution of the ecosystem, that each of these individual organizations have more or less a role to play in here that's important for us to be realizing, because not everybody has the same incentives. Dr. Apfel was talking earlier about in a for-profit and not-for-profit hospital, still the goal is to actually make some revenue. And that's what happens for our entrepreneurs who are looking to turn their product into something that's cashable, as well as for the investors. So the next category is looking at opportunity size. And so sometimes when people are trying to develop their opportunity size, they actually confuse health savings with company revenue. And that's an important thing. You may be able to say, if I do this for you, it's going to save the health system $20,000 if I use this tool versus this one, or do this procedure versus this one. And that is actually a savings in the health system, but it may not be revenue that's coming back to the company or to the investor. So if I'm an investor looking at companies, I'm looking very carefully to see what have they put in their market opportunity size, so that they have not either miscounted or overcounted. We also look at whether or not their product technology is actually protectable. So we really are looking for things that have strong IP. We want to see that at the beginning stages, they've actually been able to prototype something and get it out into a testing environment quickly. This happens to be my product in Evolution, one of the prototypes. And so you have mechanical engineers who actually want to always make it a little bit more refined, and let's change the bore angle and things like that. And you've got clinicians who are saying, let's just put it on the end of an endotracheal tube with an ambu bag and test it out and see if it's actually going to work. So that ability to be able to move things quickly out into prototyping so that people can see whether or not it's going to be useful is more important. What we really don't want to see is people who are in love with their technology, but haven't really figured out the customers well enough, and not figuring out the customers and whether or not this is something that they're really going to need and use as a super important component. So that's just my example of things that we're working on. Then we talk about people who actually, teams have come to me and said, I want a marketing sales channel or partnerships. And so I think it's very important in the early stages that as you're looking, when someone says, I want the budget to hire 2 salespeople, that you instead may say, do you really need a salesperson, or do you need someone who's actually helping do a value proposition canvas so that they can understand all of the people in the ecosystem and how these products and services actually give benefits, gains, and influences as well are getting paid. So we want to look at the competitive analysis, and everybody's seen this slide. I just put it in here as a placeholder because I think what's useful is to actually look and see whether or not the company is going to market with a rational strategy. And so we may have, I'll use the regulatory path. If you look at a 510K, that is actually a faster route to market because you actually have someone who's already out in the market and you're comparing yourself. I as an investor, I'm looking at someone, when I say, what's your predicate? And they say, I don't know what a predicate is, but they've told me they're on their 510K pathway. I get a little bit concerned that they really don't understand. But I also don't want you to shortchange by taking a pathway that's easier to get to market when you might get some benefits of breakthrough designation or a more stringent pathway, which will then actually give you the opportunity to have some back-end protection, because the first in will get a little bit longer to be on the market. One thing we don't want to see is that people are coming back for more capital, or they haven't actually planned their capital. So if we take the three patterns of drugs, biologics, devices, and diagnostics, and small molecules can go into there as well, and look at the product definition stage, the preclinical stage, and then further on, it's the FDA stage. Usually we're seeing companies at the very early stages who are actually trying to advocate and make sure that they get to an FDA clearance if they do require it. And so we're really looking for people to clearly identify, this is going to cost me this much money to get to here, and have some very significant milestones for adding value to what they're doing as they take the money. And so the worst thing to do is find that you say, well, what milestone did you get to with the $2 million that we just gave you? And they're not able to articulate how far they added value to the company. And they're not able to articulate how they actually are, how much money they need to take to the next round and then through to exit. I'd like a company to be actually showing me that they understand what their exit strategy is going to be, who they're going to go to market with, if they, because very seldom do products actually get out in the market without a partnership of some sort. Whether or not it's you're packaging your item with another, you're replacing something that's already in a kit, or you're replacing a procedure or a product that doesn't need to be there any longer. If that's quite, if it's quite aggressive in terms of your change in solution, then what we're looking for is that you actually understand what it's going to take to get you there and what it's going to take to exit in the company. And so my last slide is really talking about how we're moving from risk to value. So I think it's really interesting when someone says to me they hired 2 sales reps or they built an MVP. That really actually is not a value statement. What I'm looking for is this actually makes you this much further along in terms of your bringing value back for the company and therefore for the investor. And so I'd like to see some very strong metrics that KPIs that people have actually achieved. And Hall gives a very good talk about what metrics that one should achieve. And I'm sure that people might have actually heard him do that before, but it's a good discussion. I see there's something in the chat here.
Let me check.
Okay. Oh, yeah.
We asked people to post it. Basically, we say if you would like to ask a question, post it in the chat box. And I have some questions when you finish. Are you at the end?
Yep. So I'm just going to say thank you for the opportunity to share my thoughts.
Great. Thanks. So my first question is, what do you look for when they show the FDA path up there? What's going through your mind in evaluating it?
What's going through is it the most strategic path for the company? So I consult with the university here, and I'm part of the University of Washington Brahms guest faculty. And so when we do actually open hours where people can come in and talk to us about what their strategy is going to be, I'm looking for them to understand that what classification of drug or device that they would be, if there's something that's actually at, for example, the device that I described earlier that we're coming up is a class 3. And so I'm looking that people understand what actually it takes to develop a QMS system, to develop all the product validation and testing that's required, understand the safety and efficacy requirements that are needed, understand that once they make claims against when they submit their FDA and say, I can use this for these claims, that I won't be then later on getting a regulatory notice that says a warning letter that the company is actually using the product or service for something else. They've broadened their claims. It's okay to be coming into the market on one claim and then asking for regulatory approval to go to the next broader activity, but it's not okay to do that without actually taking through those steps.
Great. My next question is, there are many exit strategies here. They can exit after clinical 1, clinical 2, FDA approval, or after you start the business or grow the business to a certain extent. What do you like to see in a pitch with regards to the exit? Is it the earliest possible, or do you want to find the biggest value? Where do you think is the right place for a startup to put that? Because you really don't have time to play all the scenarios out in a 10-minute pitch.
No, that's true. Sometimes when people put the exits on their slides, it says to me that they're actually thinking about that. But what I want to see is whether or not you've got relationships with people that you're going to exit. So wherever you are deciding to exit, you actually have to build those relationships early, and whether or not it's with a strategic or it's with a mutual packaging where it gets kitted with something else. I'm wanting to see that someone's clearly thought about what that's going to happen, how that's going to work.
Great. My last question is, whenever I see one of these presentations, I see a very long list of intellectual property, patents, trade secrets, copyrights, and other things. And my question is how do you evaluate that strength of that patent portfolio in the good 30 seconds you have to look at it?
Well, that is true, that you can't. So what I don't think you can dive down into, sometimes people have a patent, but their freedom to operate is not clear in terms of taking this service to market. So I do think that you're relying a little bit on the company having at least a patent attorney that's actually helping to give them advice, but also that they have looked at a portfolio. So it's no good getting just one patent. You need to be actually thinking about how to layer the patent protection around what it is that you're doing.
Great. Well, with that, we're at the end of our time. I want to thank you for your presentation. Appreciate you sharing that with us today. And let's go ahead and introduce you as an investor today. Can you tell us a bit about your background as an investor?
So I have been generally group investing, and that's the way I chose to do it, because I felt that when you think about what do you do in due diligence that actually can reduce your risk. I'm not a patent attorney, and so that to me required someone who has a little bit more knowledge about that. So I actually like working with other people who can see things from a different perspective. I invest in a group. I've invested in 7 different companies, in addition to some that we just take equity in. I have physically put money into the others. I used to think I was going to mostly do it in health and life sciences, but actually I have a strawberry robotic picking company, because I believe that agricultural food security is an important thing, and I see that the difficulty with getting workers to the fields. I also have a coaching platform. When we went all virtual, people were missing that opportunity to be coached at the next stage of their role. So I invested in that. And my third thing is, that's not health, I should say, is to, this is a, I guess I'm sorry, I'm hesitating about a tiny bit about how to describe it, because the rest of my other health ones jump in. I'm in a proteomics company that's actually doing a digital platforms to make to use AI to make decisions on using proteomics and speed up the drug delivery pathway, which has actually been interesting, because as we look to bring our aerosolization to the market, it's been fun to see what's been going on there.
Great. Well, thank you for your presentation, and thank you for joining the investor panel today. Our other investor is Dr. Chris Apfel of Life Science Angels and also of SAGEMEDIC. Dr. Apfel, can you tell us about yourself, your investing, and how you approach life science?
Yes, probably in a very similar way. By the way, I liked your presentation and would love to, if I can get a copy of the slides, would be great, because this development pathway, that would actually be nice to look at it. It wasn't more for drugs or devices, but there may also be other areas on diagnostics where it may look a little bit different, but I really like the way how it was arranged. So congratulations on that. And I'm primarily...
Well, I must give credit to NIH Catalyze for that slide.
Okay, good. But nevertheless, thank you for picking that. I wasn't aware of that. So yeah, I'm the chair of life sciences at the Karatsu Forum. I have been with Life Science Angels. I have not been active with Life Science Angels recently. I've made roughly 20 investments in the last couple of years. And I'm primarily consider myself an impact investor. So I only invest in stuff that I really believe can make a real difference for our society and is addressing a really significant need. And so that's the reason why I'm primarily looking into life sciences. That also happens to be my domain expertise. And but I have done some other investments. So for example, in Soteria, which is a battery company that actually can prevent or has a technology that makes the batteries non-explosible. And so so battery fires is a real issue, and they have a solution for that. And it's faster, cheaper, better. So that's very nice. And in general, when it comes to life science investments, I'm looking at usually devices. I also look at diagnostics. I have tried to stay away from drug development, but I have also made 1-2 investments in drug development companies as well.
Great. Thank you for that. And thank you both for joining the panel. And with that, let's bring up our first presenter. I have a little bit of a change of order here. We have Adam Greenspan of Trace who will kick us off with the first presentation. Adam, if you go ahead and show your slides and kick off. For those that will be presenting, we'll have following Rejuvenation Tech, Creative Biotherapeutics, Rejuvenation Technologies, and then Scaffold Therapeutics. But with that, let's go ahead and kick off with our first presentation. Adam, go ahead. We see your slides.
Excellent. Thanks, Paul. I'm Adam Greenspan, CEO of Trace Orthopedics. With our products, tendon repairs are made easier to restore mobility for millions of patients. There is a major gap in the treatment of partial tendon tears. They occur all over the body, most commonly around the hip, shoulder, and ankle, but due to cost, risk, and recovery time, they're typically not referred for surgery until it becomes a complete tear, which can take years. Our solution is a patented trace tendon fastener system. We use what's called a percutaneous approach, which is insertion through a single small incision needle guided by ultrasound. And I have a video animation that I can pull up right here. So it's guided by ultrasound. That's the probe right there with our insertion apparatus. And then it's an implantable anchor screw and spring washer that's fitted inside of this 10-gauge delivery needle. This implant is going to come into the picture now. So the spring compresses into a washer and the loose tendon is reattached to bone without full anesthesia in an outpatient setting. And this major breakthrough makes partial tears more treatable. Current landscape ranges from minimal to more invasive. Frontline treatment, starting on the left here, is conservative, including injections, needling, or a micro burning technique called tenotomy. This is a growing market, still does not achieve actual repair. On the other side is open or arthroscopic surgery, which is a mature and crowded market, but there's nothing in between that bridges these treatment approaches. This manifests as a major treatment gap for partial tear. That's where Trace comes in with a repair product that is minimally invasive. So we see a huge market opportunity with over 31 million cases reported each year in the US. We have a readily serviceable market niche classified by cases of partial tear with clean edges, that is no shredding of the tendon, no retraction away from the injury site, And this counts for about 10% of partial tear cases, representing a $2.4 billion serviceable market. So we're starting with gluteus medius at the hip, which is essential for walking. Here's a case that started with a partial tear seen on MRI. These two images shown here. The one on the left, there's brightness pointed out at the injury site, and that's the fluid for the partial tear injury. This patient had conservative management, platelet-rich plasma injection, and physical therapy. And then after 6 months on the right, the tendon completely torn away from the bone. You can see by the increased bright fluid signal. At that point, the only therapy to offer the patient is open surgery or a walker. And these cases happen all the time, and there are low-hanging fruit. We're right at the forefront of a paradigm shift for tendon repair by performing these repairs outside the operating room in a procedure room like image shown here. We're cutting the healthcare costs in half compared to surgical cases and shaving months off the recovery time. Essentially, we're expanding the market, delivering repair treatment to patients who otherwise are not referred for surgery. So we initially proved the concept by placing our product into human cadaver, standard for feasibility testing MSK devices. Since then, we've updated the designs and developed product specifications and prototypes for more cadaver labs and animal testing, which is underway. The 3D printed delivery system seen here passed our design review. And here's my co-founder, Dr. Bill Morrison, placing the trace fastener into a human cadaver with a portable ultrasound unit for guidance. So we recently completed a pilot animal test with the veterinary school at Colorado State University, which operates a cost-friendly clinical research organization. So the trace device profile is much smaller, you can see on the top here, than the comparison washer device off the shelf. And ours maintains stability in all 3 sheep. Also repeatedly withstood almost 700 pounds of force in mechanical testing, which is over triple the strength of industry standard suture anchors. So we have robust intellectual property, patents are issued and pending worldwide on the implant, the delivery system, various improvements, plus we have method claims. For FDA, it is a class 2 medical device, so it should be 510K-able. No clinical trials as we show equivalence to existing product. Our predicate is an anchor washer system. We started the submission process, and next we'll share results from this pilot that we did in sheep and our protocol for a pivotal animal study. And that'll take us into 2024. We're on track for clearance in about 15 months with initial market launch planned shortly after in Q3 of 24. Our go-to-market strategy will be ready to fully activate on day 1 after FDA clearance with a planned alpha launch. We have a dozen commitments now from interventional hip specialists to be alpha sites as we launch with the gluteus as our beachhead. Then we have over 50 general hospitals and private practice groups lined up for our subsequent expansion phase. As we add clinical applications, our plan is to scale the sales team, begin calling on orthopedists, plus partnering with international distributors. And by then, we would envision specific medical reimbursement codes for the trace procedure. In the meantime, there are general payment codes available that can be applied to cover trace repairs, showing a table now of the ones that we've identified. And this will facilitate early acceptance by physicians, particularly interventional radiologists, will be attracted by the surgical rate associated with these codes for these procedures. We're also hearing strong interest from orthopedics as well, given that it's such a short procedure time. So our revenue model flows from selling single-use procedure-based products to hospitals and surgical centers. In the context of the overall payment scheme, trace devices will be procured by the sites of service, which will follow our guidance to obtain the reimbursement as applicable. Initial ASP of the main trace fastener kit is $800. with a COGS of 198. Some cases will use a second preloaded fastener with a similar profit margin, just over 75%. Looking at our forecast, the first revenue is in late 24 from the alpha launch. We'll have repeat users and add users and add indications, fueling a jump in procedures, pushing our revenues up quickly so that by 2025, we're profitable. We'll be repairing rotator cuff and Achilles injuries and informed by orthopedic surgeons, also used as an add-on in total joint replacement for preventing surgical complications associated with those cases. Our team covers the entire medtech spectrum with dozens of patents issued, products launched, deals executed. I'm CEO, I'm a biomedical engineer with over 15 years in licensing and entrepreneurial program management. My co-founder, Dr. Morrison, is a KOL in the field of musculoskeletal radiology. He started in the Air Force, and he's had an extensive career at Thomas Jefferson University and Hospital in Philadelphia, where he's at now. And he has a product on the market, a steerable needle. Our board chair is Paul Janeski, who runs a device incubator and asset broker firm, Hatch Medical. He is an active angel investor and a member of Keiretsu Forum in the Southeast region, and he brings over 30 years in device sales and executive management. We also have world-class advisors, and our medical advisory board includes key opinion leaders in sports medicine and orthopedics to help drive clinical adoption. Our Series A is open now. It's led by Keiretsu Forum Mid-Atlantic. It's $1.5 million at a $4.5 million pre. We're using their term sheet, so it's designed to be investor friendly. It's preferred participating equity, the one-times liquidation preference. We have just under 600,000 still open. So we're in tranche 2 of 3 with 20% warrants and then 10% for the last tranche. Our goal is to achieve a premium exit, monetizing our IP by licensing to a strategic or through an outright acquisition. We're projecting an ROI of about 5 to 16x in 18 to 48 months. And I see 4 value inflecting milestones along our exit path. First is getting FDA clearance. And then after successful alpha launch, and the usual target is 100 cases. So at that point, there is a strong likelihood of a high value license or an early acquisition. If not, then we'll open a B round to expand our indications and introduce product improvements such as surface coatings and absorbable compositions. And at that point, we'll have a fully assembled management team and our board driving Trace to an exit target nearing 150 million, based on some handsome revenue multiples that we're seeing in deal comps today. Highlighted a couple at the bottom here. Trace Orthopedics is pioneering image-guided fixation. Join us as we give people back their mobility.
Thanks, Adam. Elizabeth, you had the first question.
I do actually. So tell me, if you could go back to the slide beforehand, what is it going to take you to that next, you're raising now, but what is it going to take you to that next inflection point? So does that take you to the end of Q4 2024 at the alpha launch?
So yeah, the raise now, it'll get us to our FDA clearance submission.
So earlier, okay.
Right.
So then how much are you going to?
And yeah, so we've done it in stages to get to these inflection points. So then we are going to need some additional capital to get to market through this alpha test. We're actually working on some grant applications, but we're envisioning to do a bridge into a B round. So, and that to get us that initial first in man on the market.
Okay.
Right. Chris, what's your first question?
Adam, I wanted to congratulate you on your presentation.
Yeah.
I think it was well done, and I love the invention. I think it looks very, very cool. I have, of course, some other ideas on can you do it with absorbable material. There are some absorbable screws that are actually osteogenic. So that would be one consideration. I'm not sure whether they have the mechanical stability. Per weight, they have actually higher mechanical stability than steel, but one needs to look at their volume. What I don't know is how well this works, how large the market size is. I've actually looked up at Achilles tendon. The complete ruptures are probably between 10 to 20,000 a year. So there is a market there. The partial ruptures are likely to be 2 to 5 times higher. So there is, certainly is there a market. What I do not understand is in your solution, you actually need to have the tendon and the bone. Along parts of the Achilles tendon where it actually ruptured is actually not where the contact is with the bone. So that's kind of one of my concerns. And the same may actually be with the trochanter in the hip region. Mm-hmm. And so one of my questions would actually be, could this screw, will it really relieve pain? And could it potentially even increase the risk for a rupture? So that's still for me a question mark. I don't know. The anatomy of the shoulder is quite complex, so I'm a little bit concerned there. I do think that. So my question is, compared to the predicate, you said there is something with the washer. If we had some understanding what the market size of that company is, that would give us an estimate on what our market size could be, and our market size hopefully could be twice as long, twice as big. So my question is, what's the market size of the predicate device that's currently used for partial tendon tears?
So yeah, it's a nuanced answer that I don't know if we have time for now, but the predicate is, so there's not that differentiation between partial and full thickness tears within the regulatory history. So but you're correct. We're looking to make the repair at the footprint of where onto the bone. So rather than at the at the peak stress in the middle of the tendon. Yeah, we are treating a subgroup of those, but also correct that all tendon tears start off as partial tears. So it is a huge market. And yeah, we are looking to treat the specific injury cases that come off the bone at that enthesis site. So it's a very it's a niche application, and we but the strength that we're comparing to is it's the predicate doesn't have the market, and like we don't have the market intelligence on the predicate device because it's not used for the partial tear case, and it's
so is it used for a complete tear?
Not repaired as frequently. So yeah, that hasn't been the way that we've quantified the market. We've just tried to build up based on our network of adopters and make assumptions on how many cases that they would treat on a monthly basis. And so we've done that. We just did that for the hip franchise first. And yeah, I mean, we can talk offline about it. But
in the interest of time, can you imagine your device being used for complete tears? Why partial tears? Because that would make it simpler also in terms of estimate market size and commercialization potential.
Right. Yeah, it's
great.
Yeah, we're focused on that specific case of the partial tear augmentation.
Great. Thanks, Adam. Good presentation.
Yeah, thank you.
Thanks. And we'll go ahead and bring on our next presenter. We have Anita Davidson and Donald Davidson of Creative Biotherapeutics. Anita, Donald, if you could go ahead and launch your slides and go and kick off with the next round. those in the audience, if you have questions, post them on the chat box, and we will send you a poll throughout each one to see what specific interests you may have. Please give us your responses there.
Great. Thank you, Paul. As you said, my name is Dr. Don Davidson. I'm the CEO and co-founder of Creative Biotherapeutics. We're a biotech company that is delivering novel therapies for drug-resistant diseases. Our motto is we succeed where other therapies fail. So we're asking for $3 million to complete our phase 1 drug scale-up for GMP studies and our tox studies. After that, we'll do a second close of $7 million to complete our phase 1 human clinical study. So what do we do? Well, I created a first-in-class anti-cancer and anti-COVID therapy known as CBT-300. CBT-300 reverses drug resistance in recurrent cancers, and it stops severe COVID. There are no effective therapies right now for these patients. The problem we're going to focus on, though, is recurrent drug-resistant cancers. Currently, around 600,000 mortalities every year for recurrent drug-resistant cancers. These are the top 10 cancers in the US last year. As you can see in the white bars, those are the ones that reoccur. Now, on the average, these patients have failed all other therapies. They've had about 4 to 5 other therapies and they've failed them. So we're looking at just the ones where our therapy is shown to be effective in animal studies. And if we look at that, that's over a $21 billion market annually. And our goal is to enter the market in metastatic breast cancer in 2026, second quarter. Our solution, CBT-300, is designed to reverse drug resistance in metastatic and recurrent cancer. So how does it work? Well, we all know that cancer grows faster, has more nuclei, just stacks on top of each other. But what you may not know is that I spent 20-some years, 25 years researching cancers and how they come back. And I noticed that every time we took something, I was working at Abbott and AbbVie, to the clinic, we were always getting the patients had recurrent cancer. They had drug resistance. And those are not the ones, the cells we had. So I started studying drug resistance years ago before it became a thing that now most people are doing, most companies. And what I found is all drug-resistant cancers that I researched, all of them, put a protein on the cell surface called GRP78. It's a stress protein. When they do that, they become very resistant to immunotherapy, chemotherapy, radiation. And so what our therapy does is I designed it to take that protein off the cell surface. And when you do that, now all of a sudden those tumor cells become sensitive to immunotherapy, chemotherapy, and radiation. So now we're revert back to a normal phenotype. So our data is a platform therapy, CBT-300, cures recurrent drug-resistant cancers by reversing the resistance, reversing the immune resistance, and creating normalized cells. So does it work? On the left here are 4 different cancers in mice, put in mice. These are mouse cancers, triple-negative breast cancer, renal, melanoma, and lung cancer, compared head-to-head to the standard of care Keytruda. And we just did survival in each one of these. And you can see that CBT-300 increases survival in melanoma 63%, where Keytruda had no effect on melanoma that didn't have PD-1. Same thing with lung cancer. We saw better survival, renal cancer, and about the same survival in breast cancer. Although in breast cancer, PD-1 is only expressed in about 20% of the model, and ours is expressed over 90%. To look at, does it work with chemotherapy? What we did in this study is you can see the tumors on the bottom. And you can see that controls are very big, they're very bloody, there's angiogenesis. However, when you add doxorubicin, you do get a smaller tumor, but when you add doxorubicin at a lower dose, with our therapy, you get a much smaller tumor. Again, it shows that we can reverse that resistance in these tumors. And finally, the new thing that people are doing is doing spheroids. And so on human cells, you take drug-derived or patient-derived tumor cells, grow those in spheroids because it mimics a tumor with hypoxia and low glucose. When you do that, you add your therapy to it and see if you can change the spheroid. When we did that, you can see that doxorubicin really didn't have much effect in pediatric brain, breast, or ovarian cancer. When we added CBT-300, we could shrink the tumors dramatically, and in combination, many of the tumors just completely were gone. So our indications we want to start with are triple-negative metastatic breast cancer. We want to be in the market in the second quarter of 26. We had a really good friend of ours that died of metastatic breast cancer with 4 kids and left a very outstanding career in music. And so it's one of our passions that we want to go forward with at this point. So what about competition? Well, for metastatic breast cancer, there are competitors. All of them are showing resistance at this point, at least 80% resistance. CAR T cells has not shown any effect so far, although it's in a phase 1 study. Whereas our therapy, CBT-300, is a much lower cost. It is non-toxic. In all the studies we've done with mice, and so far we've not seen any toxicity at all. We haven't seen any resistance. I believe that GRP70 on the surface is essential for resistance. It can be used in combination with all the other therapies to make them better, since we will reduce the resistance and actually decrease the immune resistance too. Our target is expressed, like I said, on almost every single resistant cell I've ever looked at, and it can be used for multiple diseases. So our advantage, well, years ago I read that it takes 10 people, 10 years, and $100 million to find the target, to validate the target, to find the drug, to optimize the drug, to do proof of concept. We've done that with 2 to 3 people, 4 years and $2 million. So we've been very cost effective and very focused on what we've been doing. We have a strong intellectual property position. We have a composition of matter patent. We patented 5 different compounds, and we're going to put in a second patent to cover even more compounds to try to protect that property so nobody can hit that part of the molecule that we're hitting. We also have international and application use patents for virus and some other diseases which we are targeting. So the risk and mediations. Will it be toxic? We have not seen toxicity at all in the mouse studies. And the reason I don't expect toxicity in humans is because GRP78 is 99.4% homologous, exactly the same sequence in all species, no matter what you have. Efficacy, we've shown good efficacy in animal models. Human testing, we've done those spheroids. So I know for human cells that so far in the publications, if you show spheroid results, it will work in the patient. So it's shown a really nice result. Oh, the team. So my name is, Don Davidson. I have over 33 years of drug discovery and development from bench to the clinic. I moved 4 drugs from the clinic, from the bench all the way to the clinic and to marketed. Venetoclax is one of them. Kinlytic is another one. My co-founder, Dr. Anita Davidson, has run a healthcare clinic for over 25 years. She's founded 4 successful companies and is a true expert in project management and revenue generation. We have a model that's very decentralized. We have our CROs doing the heavy lifting for making our manufacturing, our distribution. We have Syneos Health that does our regulatory. We have legal team. And we know we'll have to bring more of this talent inside when we get into marketing as we get further along. But right now we can use their talent and their expertise to do these same things. Our projected return on investment is 22 times in 5 years once we hit the market. We have several exit possibilities. With the environment the way it is, I'd hate to pick one. So we know that phase 1, phase 2, or even a BLA, we could exit with any kind of a partnership or any kind of licensing deal. And we expect to go forward with that in the future. So our ask is $3 million to do our phase 1, and that will do our estimates from the rat tox and our drug scale up. Once we get that done, we'll apply for the IND, and then go into phase 1 clinical studies from there. We have raised non-diluted and diluted funds with the National Science Foundation and have over $2.1 million of raised funds. So why it matters? Well, we have a strong intellectual property position through 2041. Our investor value, we expect around a 22 times return on investment in 5 years. We have one therapy for multiple diseases, multiple cancers. The product is safe, non-toxic with no side effects so far in vivo. It's easy to produce, it's very stable. And our competition so far, just a head-to-head, although we can make their drug better, we have shown superior activity in several different cancers. Thank you.
Great, thanks. Dr. Apfel, why don't you take the first question on this round?
Okay, well, thank you for your presentation. What you promised sounds really impressive. I would love to understand whether your response is a response to spheroids or a response to primary patient tissues.
So the primary patient tissues we have for DIPG for that tumor is primary tissues. We're just taking the tumor's cells out of those tissues and growing them in spheroids.
So
And
it's
we get a
primarily
very-
patient-derived spheroids. Mhm.
Yes. Yes.
Okay, and that's for metastatic breast, for triple negative or metastatic breast, or?
We haven't done the triple negative. We did a cell line for triple negative. We are getting those patient-derived tissues to do the triple negative breast. We have done at least 7 or 8 DIPG brain tumors right now, definitely from kids and other stuff, but we haven't done the metastatic breast yet.
Okay, but you have done this with primary tissues from patients with brain cancers.
Yes.
Okay. It would be nice to understand a little bit the mechanism of action at some point.
I would love to talk to you about that anytime.
Yeah, we should probably have a separate conversation on that.
That'd be great.
What was to me a little bit got me off the wrong foot was the first thing you were doing is asking for money. And I didn't even know what you're doing. You had a very general statement of, we make curable disease curable. And I want that many dollars. And I don't even know what the pre-money valuation is. And I didn't even know what you're doing. And then you were saying, and we can cure cancer and we can also cure COVID. Without the background that you have, it didn't sound credible. Right? It sounds, so one of my questions would be, how much money have you put in yourself? I mean, you're apparently in this together. Can you comment on that?
Yes, sure. So we put in 200,000 so far between Anita and I.
Okay. How much other investment, and I'm not talking about grants, how much other investments have you raised so far?
We've raised $825,000 from angel investors, initial seed investors.
At what
It's been
pre-money
word of mouth.
valuation? At what pre-money valuation?
Currently, we are in process to start due diligence with Keiretsu, Mid-Atlantic, and Southeast. And we have been our valuation is set at $20 million, is what was determined by the head of the forum.
We probably should run this through our smart cap table and to take a look at that.
I was thinking a waterfall cup, but yeah, it would be a good idea there.
Absolutely.
Yeah. We understand that valuation, especially this year with everything changing, is a bit of a moving target. So we've consulted multiple experts, and it's still pre-money is still very difficult to try to value. So we'd appreciate any additional input.
Yeah, we have I think I have something. We should have an offline discussion to go run through the numbers, because I think that there could be a happy medium. At what pre-money valuation did the 850K have those been invested, or were these safes where there was no cap?
No, they were invested at 5 million. The original 100,000 was actually more gift. We didn't even have a valuation because we had not done any in vivo studies, and so we weren't sure whether we were going to have any efficacy. And so then the next round was at the 5 million, and that was prior to the bulk of our animal data.
Okay.
So your animal data has brought you to a 20 million on the strength of what you've done. And is that also because you've expanded your patent portfolio as well?
That is it. The patent portfolio, the animal data, and the money that we've raised. So they use a 9 times return on how much money you've raised total. And so that's the average that they told us they were going for. So that's how we got the 20 million.
So out of the 825, you said 100 was a gift at the beginning, and then you put in 200?
Yes. The 100, it started as a gift. We did equity. We did go back and give those initial investors equity because it was the right thing to do, because they were stepping up right at the beginning. And so we weren't going to leave them behind.
So have you seen the term sheet for Keiretsu?
Yes, we have.
We have negotiated it with Howard for Keiretsu. Yes, it's not been implemented yet. It's in his court. But we have had a couple of different conversations and negotiated the terms.
And
No,
you've been careful to protect those early investors, and that'll happen for the next group of investors?
Yes, yes. So absolutely. Those terms are in there. And we would do that regardless. Our prior Keiretsu term sheet was also making sure that we were not over diluting or over in any way harming our initial investors. I think it's an important piece.
Great,
Thank you very much for
Great.
that. And it was fascinating.
great. Thank you guys for great questions and a great
Thank you.
presentation. We're going to move on to our next presenter now, Glenn Markov of Rejuvenation Technologies. If Glenn, you go ahead and put up your slides, love to see your presentation next.
All right, great. Hi, guys. So I'm Glenn Markov, co-founder of Rejuvenation Technologies, and we develop messenger RNA therapeutics for regenerative medicine. The founding the company was formed during mine and John's research at Stanford, where we were both interested in intervening in the aging process, and thus having the potential to impact many diseases of aging, where the single biggest risk factor is just your biological age. So we thought, and there's good modeling to support this, if you can intervene there, you can have a big impact on the broader healthcare system instead of going after each disease one at a time. So one of the fundamental aging clocks that we know about are telomeres, which are the protective caps of DNA. They're a repetitive DNA sequence, and they allow the linear chromosomes that we have, to form a loop at the tips of the DNA. And when telomeres divide, when cells divide, telomeres shorten, and they can shorten for other reasons as well. And so as we get older, our telomeres are gradually shortening. And the way they function as an aging clock is they end up triggering other hallmarks of aging, like the ones shown here, such as classically stem cell exhaustion, so that the lack of telomeres results in cells arresting, so they're no longer dividing, they can't regenerate tissues. You also see things like genomic instability, so chromosomes fuse with each other. You can see senescence, of course, and then others. And so this has been very well studied in the context of human biology and animal biology. And it's known that, for example, in mammals, the rate at which telomeres shorten is a highly correlative predictor of lifespan for a given mammal. And so it makes sense that telomere extension would enable regeneration. The reason being that when cells are dividing, they're able to replicate and repair tissues. And if telomeres are short, cells can't divide. The strategy that we're employing as a company is to develop a drug that allows us to extend telomeres and bring the patients that we're treating that have short telomeres into what we call the Goldilocks zone. So the telomere length varies between something closer to 10,000 bases during the beginning of adulthood to 5,000 bases towards the end of life. And during that time, of course, the telomeres not only shorten, but there's a fraction of telomeres that becomes dysfunctional. The Goldilocks zone is quite big, so our challenge of bringing patients into that Goldilocks zone is not, that's not the challenge here. The technology that we have is a synthetic messenger RNA encoding for telomerase, which extends telomeres. It allows for rapid targeted telomere extension. The reason being because the messenger RNA codes for the telomerase protein, and it's been honed by billions of years of evolution to perform that exact function. The telomerase RNA and the protein and the subsequent telomerase activity are very short-lived, providing temporal control and giving us an excellent safety profile. We deliver the messenger RNA with lipid nanoparticles. We have a lung and a liver lipid nanoparticle that target those diseases, and we're developing therapies for specifically treating diseases of aging within those tissues. The initial data was published at Stanford, and where we showed that telomere extension rewinds the clock substantially. In fact, a single dose corresponded to approximately 500 bases of telomere shortening that was reversed. 3 doses correspond to about 1,000 bases. During human aging, telomere shortened by about 50 bases a year. So this corresponds to about 20 years of telomere shortening. And when the cells looked younger as well, there was a reduction in large cells and senescent cells. This is the best in class approach to telomere extension. Telomerase DNA has been studied for decades as an approach to immortalize cells in culture, and thus poses a similar risk when treating human patients. The small molecule approach has also been attempted for years, and small molecules are good at inhibiting certain classes of proteins. In this case, the goal is to activate a gene that is typically not on, and so that is usually out of the realm of small molecule biology. The formulation process follows standard mRNA lipid nanoparticle procedure. We mix the messenger RNA with a cocktail of lipids and do a series of purification steps to arrive at these lipid nanoparticles where the messenger RNA is encapsulated. These look like kind of a milky white substance. They're about 50 nanometers, like I said, and we delivered this intravenously. And depending on the cocktail lipids we use, it targets either the liver or the lung. In the case of the liver LNP, we're using off-the-shelf LNP from a partner company. And in case for the lung LNP, we've developed the lipid cocktail ourselves and tested it in multiple animal models, including primates, to show that it's safe and delivers the RNA to the proper location. Here we're using messenger RNA encoding luciferase to make the organs glow, and you can see the organs that are glowing receive the RNA, and the lung is glowing broadly throughout, and most of the signal is coming from the lung. Now, when we look at the cells that are being transfected, you can see there is quite a few cells, almost over 95% at the highest dose. We've done a number of safety and efficacy studies in animal models in the last few years. This is because we wanted to test both in the liver and the lung, and using short telomere mice, the effect of the drug. We find a dramatic reduction in lung fibrosis, and improvement in the lung forced vital capacity. So the functional improvement in the amount of air that the mouse can breathe to restore it back to baseline. And this is used in clinical trials as the endpoint for drug approval. We've also looked at features of the liver, including liver fibrosis in multiple models, where we see dramatic reduction in liver fibrosis, inflammation, and DNA damage. We also have examined safety on a number of different axes, including drug clearance, which is quite rapid. We've looked at tissue histology and had a board-certified pathologist take a look and report findings, which included no moderate or severe findings, no findings at all in the lung, and mild infiltrate in the liver and kidney. We see a low immunogenicity. So after 2 high doses, we go back and check the levels of cytokines, and we see that they're near baseline levels. Our plan is to continue to advance these 2 programs in age-related diseases, such as pulmonary fibrosis and liver fibrosis, through multiple milestones, including phase 1 clinical trial readout. And so our next steps are scaling up the drug manufacturing and doing our GLP toxicology studies. Programs that have shown some promise in the clinic are usually valued in the billions of dollars. And so I think this makes it an attractive path for investors. This includes both publicly traded companies, so many biotechs go public. So there's many small biotechs, public biotechs on the market. Pliant and Vicore, 2 examples of ones who have a drug in clinical trials. Again, once the drug is approved, the value goes even higher. But typically, there's partnering acquisition that happens before then, such as in the case of Galapagos or Promediair. The current round is $2 million, of which $1 million has already been invested by a lead investor, Merchant Ventures. We also have had 600K in angel follow-ons and additional folks doing diligence at the moment. The terms are Y Combinator post-money safe with a $48 million cap, 5% discount. We also have 2 large grants coming in to support the work, both from California for the pulmonary fibrosis program and from the NIH for the liver fibrosis and alcoholic cirrhosis programs. And our use of funds would be to scale up the drug product manufacturing, including the RNA LNP, to make it similar to what we would use in our clinical trial, and do our good laboratory practice toxicology study in rats and protect the IP that we've built. This is that IP or a subset of those. So we have the original IP on use of telomerase messenger RNA for telomere extension, which has been allowed or granted worldwide in all the major jurisdictions. And we also have pending applications for treating liver diseases and also the lung targeting LNP. As we're progressing the lead programs forward, we also keep in mind that the telomerase is really a platform technology, a new mechanism of action for treating age-related disease. And this was a review taken from a review published in Nature Cell Biology outlining the different organs and tissues and affected diseases that could be addressed by a telomere extension technology like ours. We have a very strong team of both in the founding management team and as well as our advisor and clinical boards. John Ramirez and I did our PhD work at Stanford, and John's was in neuroscience and mine was in genetics, where we also went through the SPARK program in Y Combinator. Susanna Tulach is Director of Scientific Research, and she has experience managing a multi-billion GMP production environment. And we have advisory board members for each vertical, such as lipid nanoparticle manufacturing, regulatory, mRNA production, and toxicology. So as a summary slide, the company is founded on several primary technologies, including the use of telomerase messenger RNA for telomere extension. We've had multiple successful recent milestones, including non-human primate data, excellent safety and efficacy data in multiple models of pulmonary fibrosis and liver fibrosis. We've met with the FDA for an interact meeting to discuss our technology and receive feedback. And our upcoming milestones include the IND-enabling toxicology studies that we would need to do a phase 1 clinical trial. We'll be working on a Series A financing beginning later this year. And we've had a number of seed investors that supported us, including Y Combinator, the first one, who put in $1 million, and as well as Coastal Ventures, who led our initial seed round, Shanda Ventures, Asymmetry Ventures, Longevity Tech Fund, to name a few. Thank you.
Great. Elizabeth, what's your first question? You're on mute.
Sorry, I just was switching. Tell me a little bit about your studies. You've done it in mice. You're going to primates, and then you're going back to rats. Did I follow that correctly?
Yeah. So it's a little bit. Thank you for asking that. It's a little bit tricky to follow. So let me just pop up some of our primate data that we have. I'll share that. So we've done a very preliminary pilot study in primates looking at where our drug is going. So you can see primarily it's going to the lung. We've measured something like 15 tissues, and we're processing the additional data from that study now. So this is sort of the quantification, the distribution of different tissues we've measured, and the signal. So it's primarily going to the lung. We measured 2 different doses, a low dose and a high dose. And so but this is a study that is, again, preliminary, not for GLP toxicology. Right. So I formulated this study myself and sent it out. The next steps, like you mentioned, would be the rat and primate, which is the standard toxicology studies we would do.
You were trying to get some early signal.
Sorry, what?
I just said you were trying to get some early signal. That's why you did
Yes, that's right. That's right.
that.
Great.
Okay.
And Dr. Apfel, what's your question?
Well, first of all, sounds like an interesting solution for diseases that currently don't really have a solution. I think for both liver fibrosis and lung fibrosis, there aren't any effective therapies around there that I'm aware of. It may have been changed in the last 5 years, but I'm not aware of any that. And those are serious diseases. So I'm of course intrigued. The valuation, of course, is a little bit high. I think it was smart to point out that the exit values can be proportionally high, but one actually needs to look at the math. It does look a little bit too late of a stage for me in order to really make an impact or getting invested there.
Great.
Yeah, it's a fair point. I would just say, of course, if SAFE is a post-money cap, so it's effectively investing somewhere in the mid-30s in terms of the million valuation, which it could, of course, is still high, but it's that would be the effective if you didn't have the anti-dilution protection that the SAFE provides. So it's a little better than it looks, but nonetheless.
How much have the founders invested?
So the founders, after the Series A, will have about 10% each.
How much have you invested in cash?
Oh, personally?
Mm-hmm.
Well, as a grad student, I've invested all my time in the last 5+ years.
Okay. Are you currently drawing a salary?
Yes.
How much?
Just over 130.
Okay.
Great. Thank you guys for the questions. We're out of time now. With this, let's go and move to our final presenter, Jeff Carter of Scaffold Therapeutics. Jeff, if you go ahead and show your slides.
Okay, thanks everyone. I think probably a couple of people, including Dr. Apfel, were on my last presentation last week. So we have made some changes to the deal structure. So I'm glad to see you, Dr. Apfel. Hopefully you've got some more feedback for me that I can take action on. I'm the founder and CEO of Scaffold Therapeutics. We are also developing mRNA therapeutics, but we're developing small molecules that actually modulate the downstream biological activity of specific configurations of mRNA. So the mRNA space is pretty hot right now. As you can tell, we've got 2 presentations in a row on mRNA. And the type of mRNA dysregulation that we're talking about is methylation of mRNA. And dysregulation of that methylation is really associated with a number of serious diseases in almost every therapeutic area. So alongside of just being a small molecule drug discovery outfit, we are deploying generative AI across all of our activities. The other day, Dr. Apfel actually had a question for me that I only partially answered, which basically was whether or not this is worth doing, or at least that's the impression that I got of the question, and whether or not AI-designed drugs can get into the clinic. The first one was from In Silico Medicine on November 30th of 2021 in phase 1. And that same molecule went into phase 2 on July 29th of this year, so just a couple of weeks ago. So we believe in the technology stack really strongly. We've deployed it across our small molecule discovery operations, and we're developing a phase 2 to address some biomarker identification and translational challenges. So when we look at what an mRNA activity imbalance can trigger in terms of disease state, really a lot of this is associated with stress response of the cells and stress granules specifically. And so on the one side, when you have let's say a weaker stress response, you can end up with some of these classical diseases of aging, like Alzheimer's, dementia and Parkinson's disease. These are really large population diseases that are incredibly expensive to address from a clinical development standpoint. So we're pursuing partnerships in that area. And then we're executing a strategy to get to the clinic in oncology, specifically liver cancer, with a few other cancers in our pipeline as well in terms of indication discovery. So again, these are really large markets, and we didn't just pick them because we followed the data. And so we'll talk about that. I think the one thing on this slide actually that kind of surprised me, I didn't realize that liver cancer only had a 20% roughly 5-year survival rate, and that it was such a serious malignancy. I think almost everyone knows the Alzheimer's numbers off the top of their head. So how does the data tell us that these are the areas where we should be looking? We're trying to reduce the activity of a specific gene called METL3. And METL3 is strongly associated with amyloid beta in the brain, and it's also strongly associated with liver cancer in humans and with negative outcomes of liver cancer in humans. So we're reducing METL3 activity by about 80% in cells. That's an in vitro assay on the Alzheimer's side in a relevant cell line. We do have a decent amount of in vivo data in the liver cancer space, and I'm showing the tissue distribution data there, where you can see that our molecule accumulates significantly in the liver. So when we talk about the competition, you'll see there's a lot of traction right now in the clinic for liquid tumors, leukemia, lymphoma, and some solid tumors. But you can see with our distribution in the plasma, we wouldn't do very well there. So we are looking at liver, and how do we know that we actually have a valid hypothesis here? There is some literature that gives us precedent. So in the lab, you can actually turn off genes in animals. And so typically this is frequently done in academia. So on the left side, you can see a mouse model where the METL3 gene is left intact in the black bar, and you can see a significant accumulation of amyloid beta plaques in that model. And when the METL3 gene is turned off or silenced, you can see that those plaques are dramatically reduced in vivo. And then in the liver cancer model, similar. This is a subcutaneous injection of tumor in a mouse. And you can see when the METL3 gene is knocked out, in the right bottom image, the tumors are really eradicated, and some of them disappear almost completely. So we have an in vivo study going on right now to replicate this behavior with our pharmacological inhibitor. Where did all this come from? So early on in the history of the company, really in just the first few months, we identified Amedeo Kaflich at the University of Zurich as a guy who was doing some really interesting work in X-ray crystallography and small molecule development for these so-called M6A marks on mRNA. So we licensed a catalytic inhibitor, which is the middle patent in the table, and we co-invented these other inventions with the University of Zurich. We do expect additional filings this year, both on the generative AI side and on the chemical matter side. As anyone who's been paying attention to mRNA knows, there's a lot of money flowing into the space. We do have early conversations on partnerships going on with every logo listed in the industry box. Again, these are early. I think the most well-developed is Sanofi. We talk to them almost once a week on the partnership side. And of course, for these larger companies, if we get significant traction on the oncology side, an early partner would make an ideal acquisition channel for us, strategic acquisition. We're also aggressively applying for as much grant money as we can get, and working with all of these academic partners at some level as well, whether we have participation on the SAB or we're actually doing work with them directly. So this is probably my favorite slide. One of the reasons why in an earlier slide I said we're aggressively pursuing partnerships in neurodegeneration is because as far as we know, we have the only brain penetrant metal-3 small molecule inhibitor that is available right now. So we think that there's an open net for a partnership, probably for the next maybe 6 to 12 months. So I'm pushing on that as hard as I can. There is some competition in oncology. 858 Therapeutics is in San Diego. They're about the same level of development as we are. Their slide deck seems to talk mostly about colorectal cancer. Storm Therapeutics has an small molecule metal-3 inhibitor in the clinic in phase 1. Their first patient was dosed in November of 2022. So we expect the data out from that study probably in Q4. Most of their literature and other data point to leukemia and lymphoma. That looks to be a basket trial to see where they can get some signal. And that's great for us because it proves that there's, if the data come out strongly, it proves there's a tox window for these inhibitors. So we're excited about that. We think it will drive value for the company. In terms of the team, I've built and sold a company before, also in the life science space. It was a software company called ArcSpan. We sold that to Bruker in 2019. We bootstrapped that and operated the company for 8 years. Just this week, we named an interim CSO who I'm pushing to become our permanent CSO, Renee Lemieux. Renee has an enormous amount of experience in drug discovery and has participated in value-adding programs at a number of different companies. Jeff Hickson's leading operations. He's always employee number 1, 2, 3 at successful biotechs that grow rapidly, and he's just great at making sure that every dollar goes as far as it can go with our partners and our CROs. Gordon Saxty is running chemistry for us. He's a great med chemist and has put drugs on the market before. You can read our advisors. They're great advisors. I will point out Wasim Malik on the lower left. He's on the board. His fund, Yaso Ventures, did lead our pre-seed round, which I'll talk about right now. So we're raising $1 million. You can see the specifics of the raise on the left-hand side of the slide. I've got 15% of that money and 25% committed. We are seeing a lot of new open interest, even just this week. So I'm really excited about that. With that money, we will close a partnership, which I think we can do this year, staff a CSO and head of biology, and secure some non-dilutive grant funding. On the product side, we will nominate a development candidate in liver cancer, run an in vivo study in Alzheimer's, and we've already started bringing our first pipeline asset up towards lead optimization. We're running a catalog screen right now. We've raised $2.2 million to date, led by the management team and Yaso Ventures and some friends and family money. I've put in 10% of that money by myself. Nobody gets paid on the team, and our burn rate is, it's really about 100K. It went over 100K 1 month, so I rounded up and say that my burn rate's 125K. We think that in 5 years, we can get about a $200 million exit. I think that's pretty conservative on $25 million of total money raised. We're really doing RNA epigenetics, and the two comps that I have are DNA epigenetics companies. RNA epigenetics is a pretty new field. You can see that these valuations for Constellation and Epizyme are very different. This is market conditions. Constellation exited in 2021. Epizyme exited in 2022. So, you know, I came to the low end of the range here to make sure that my estimates were believable. So let's see. That is pretty much everything today. I'm really interested to hear everyone's questions.
Great. Thanks, Jeff. Dr. Apfel, what's your first question?
So the first question is, would you have time for a deep dive?
Absolutely. I love talking about this stuff. And even though I'm not a biologist, I've learned a huge amount about the biology in the last 18 months. Anytime.
Yeah, I do think this is it's a new evolving space, and it's a very active space, and the whole mRNA therapies are really are interesting. The question is actually, how does it materialize? I always get a little bit nervous when when I hear of panaceas, when suddenly you have neurodegenerative diseases and you have cancer as well. In terms of, for me, it's often more credible if you focus on one thing where you feel you have strong data and you stay there focused, and the other one, keep it low so that you don't get distracted. The valuation for the capital that you're raising seems fairly high. We should talk about the smart cap table and what the investment terms could be, provided that based on the preliminary data I have, I'm optimistic. So the neurodegenerative disease is really a very, very important topic. Much more money goes into cancer, but if you look at the impact for the society, the neurodegenerative diseases, at least as big, if not bigger than cancer. So having something that can work there is always worth to take a look.
So just in response to that, we're staying very focused on oncology. The literature around neurodegeneration is fairly strong, and with some lightweight BD outreach, I've gotten really good responses from potential partners. So I think those potential partners probably have a hypothesis in neurodegeneration. And obviously it's an extremely heavy lift in terms of clinical development. So I think for our size right now, all of our execution is focused on oncology, but we really want to push the BD angle on the neurodegenerative.
Okay, as long as you're focused and there's a good rationale for this becoming a successful compound.
Great.
I think there is. I'll be in touch.
Thank you. Elizabeth, what's your question?
Well, that was when you go after two things, it's nice to be able to get a little bit of solid validation before you, because then you can use some of the money from that to be able to fund some of your other things as well.
That's exactly what we're thinking, is if we can get a decent upfront and generate some non-dilutive money, that would be exceptional.
Great. Another question from the audience. What's your investment tranche size?
This tranche is 1 million. I'm trying to raise over the course of the next 12 months, probably $5 million.
Great, thanks.
And where
Elizabeth?
will that get you to?
Oh, $5 million, we should be able to get at least into phase 1. And I think if we're really smart, we can get to the other side of phase 1.
Yeah,
We'll have to raise some money into the phase 1 in order to keep going after that. But I think that money will get us all the way through phase 1.
Thank you.
Great. Well, thanks, Jeff. And that brings us to the end of our time. I want to thank our panelists, Elizabeth Cross-Nichols and Dr. Chris Apfel. Thank each of our panelists as well for the great presentations. Those in the audience, we polled you. If you had any specific questions, we'd love to answer those as well. We'll set up introductions afterwards to carry into deep dives and answer any more questions. We're at the end of our time. So with that, we'll go and close it out for now, and we'll see you guys next time. Have a good one.
Thanks very much.
Thank you.
Good luck, companies.